Hi everybody, my name is Ocampo, and this is the 6 students that worked with me in this summer project. So hi everybody, welcome to JBay, which is short for Joint Bioenergy Institute. JBay consists of Lawrence Berkeley National Laboratory, Sandia National Laboratory, UC Berkeley, UC Davis, the Carnegie Institute of Science, and Lawrence Livermore National Laboratory. So the main focus of JBay is to improve the conversion of cellulosic biomass into biofuels. Cellulosic biomass is the main component of plant, and according to the U.S. Department of Energy, cellulosic biomass is the fibrous, woody, and generally inedible part of the plant. So JBay is split into 4 departments: feedstock, deconstruction, fuel synthesis, and technology. So the feedstock department focuses on engineering plants so they can be broken down easier into simple sugars. The deconstruction department focuses on improving enzymatic breakdown of cellulosic biomass, which that's us. Fuel synthesis focuses on creating new biofuels using synthetic biology, and the technology group focuses on building new technology that could further help the overall research. So fossil fuels. As you guys can see from this graph, the United States uses more fossil fuels than any other country in the world. And also, the United States emits the most carbon dioxide than any other country in the world. So, the carbon dioxide that is released, only half of it can be absorbed naturally, and the other half goes up into the atmosphere, which helps global warming. So, according to Dr. Jeff Dukes from the Carnegie Institute of Washington and Stanford, it takes 20,000 kilograms of ancient plant material to make 1 liter of gasoline. So as you guys can see, we are wasting more fossil fuel than we are actually creating. Therefore, we need a new source of transportation fuel. So we have cornstarch. So according to the Department of Energy, the United States, the primary source of biofuels in the United States is from cornstarch. So as you guys can see from this picture, cornstarch can be broken down very easily. Amylopectin, which is 75% of cornstarch, is highly branched, like this picture shown. So whenever the enzymes come in and try to break it down to turn it into simple sugars, it attaches to the ends of this amylopectin. And since it's highly branched, it has a lot of ends to grab onto, so it breaks it down easier and faster. So the pros and cons of using cornstarch. So using cornstarch-derived ethanol reduces greenhouse emission by 21.8%. And according to Iowa State University, it reduced gas prices by 20 to 40 cents. And as I previously mentioned, you can break it down and convert it to biofuels easily. The disadvantages of cornstarch is that the cultivation of these fields that we need, it increases the CO2 emission of carbon dioxide. So also the cultivation of these fields uses a lot of water, and it uses a lot of fertilizers. We can't transport we can't transport biofuels by pipes. We have to truck it. We have to transport it by trucks. And the main disadvantage is that it causes hunger in many developing third world countries that need this food. So this picture just explains how corn prices are going up, and the stomach of this little kid is going on empty because the foods are too expensive. So this brings me to cellulosic biomass. So cellulosic biomass is the main component of plants. It is the most bountiful waste product in the United States, and the current supply from agricultural and other resources is about 1 billion tons a year. So large amount of the plant cell wall are composed of lengthy chains of sugars, or also known as polymers, that can be broken down into biofuels. So as you guys can see from this picture here, cellulosic biomass is composed of three main components. It has cellulose, which is yellow, hemicellulose, which is blue, and lignin, which is orange. So cellulose is a really hard material to reach because it is embedded by hemicellulose. And hemicellulose is linked to lignin, and lignin acts as a barrier to protect hemicellulose and cellulose from enzyme attack. So cellulose is the most bountiful, overflowing biological material on Earth. So converting cellulose into biofuels has been an important notion in recent years, but due to its structure, it is really hard to reach it. So here's another reason that it's so hard to reach. As you guys can see from this picture, the monomers that make up these chains of cellulose are stacked on top of each other. So this makes cellulose a tough and largely insoluble material. So this makes it difficult for enzymes to go in there and break it down. So if you compare cellulose to cornstarch, cellulose takes 7 days to convert into biofuels, and cornstarch takes just 2 days to convert into biofuels. Also, it currently takes 100 times more enzymes to convert cellulose to biofuels as it does to catalyze the same amount of cornstarch. So this is a picture that shows the whole cycle that we are working with. So first we start out with biomass, which is trees and plants. We harvest it, then we add acids and heat it up to make the cellulose accessible to the enzymes. So then after we get the cellulose, we attack it with enzymes, and the enzymes turn the cellulose into simple sugars. The simple sugars are inserted into microbes, which make biofuels. These biofuels are then inserted into our cars, trucks, and airplanes. And an advantage of doing this is that the carbon dioxide that it releases, it collects it from living plants. So when we burn it, it only releases the carbon dioxide that is already in the environment, instead of adding more carbon dioxide from the ground like fossil fuels. So since our current methods are not as efficient we are searching for unknown cellulose-degrading enzymes from the environment that could help us do this process in a faster way. So our whole summer project focused on this part of the graph, which is breaking down the cellulose with enzymes and turning it into sugars. We focus on improving the deconstruction process of this picture, deconstruction, which is this part. So the first step of our process is to isolate the cellulose-degrading bacteria that we want to get a single colony. So I'm gonna pass it down to my partner Ian, which he's gonna talk about it.